I have a 12 o'clock, but I can be late.
Sure, okay. What best to crack on?
Actually, the whole room has the same 12 o'clock, so we'll all be late. Don't
Pro-
worry.
What's Procan anyway? Why is it important? Why is it important? How's it work? I mean, we were doing better over there than we are now.
Or from the beginning,
Yeah.
Procan is it's a dream. What we've decided to do is collect.
Look at the eyes.
What we've decided to do is collect as many cancers samples as possible from patients around the world and analyze the proteins in those cancers in a really high throughput manner, in a way that's not been done before on this scale, and use the data to try to understand, using machine learning, any algorithm that we can develop to try to make predictions from the proteins in the cancer. So first, we make predictions globally from, say, colorectal cancer, neuroblastoma. What general principles can we glean from the data? And then in the longer term, it goes from the broad 200, 300 patients to the specific. We want to be able to hand the oncologist, the treating clinician, information for the one patient in front of them so they can make actionable decisions as part of their treatment decision-making process. So that's Procan from beginning to end.
So it's collecting samples of people. It's not just cancers? Or is it just cancers?
Procan only works on cancer. And we don't collect, our collaborators do. We rely on a network of international collaborators all around the world. They've been collecting specimens, samples from their patients for years and years, sometimes going back 40, 50 years. And they collect the tumour itself, and if they can, they collect the relatively healthy tissue next to the tumour. So we have a tumour and a so-called normal adjacent tissue. So we compare those two sometimes. That's less critical for decision-making because a pathologist can do that in a couple of seconds under a microscope. They can tell a tumour from a not tumour. But we're trying to map the proteins that distinguish the tumour from the non-tumour. The more important thing is the samples that come in from our collaborators have to be processed, they have to be digested, and the digest has to be cleaned up. If we don't clean up the digested protein, we can't feed the mass spectrometer, it gets clogged. So there's a lot goes on with the sample prep, the pathology of those samples, the imaging of the pathology slide. A slide is taken of each tissue as well, and the slide is stained so we can have a pathology report on what the tumour looks like to a pathologist. There's an awful lot goes on behind the scenes. If I step back, getting the samples is a massive task in its own right. It can take anything from 6 months to 6 years for a collaborator to put the right cohort being 100 to 500 patients together. The right samples are necessary for the study in the first place. That's a long process for some places because we need the clinical data. We have to know the outcome of every patient. So it's all what we call retrospective. We know what the patient's got now, but we need to know what they were treated with, whether they survived or not survived, and how long they survived, whether they had a recurrence or not. We need that information, not just the tumour sample. And that's what Procan puts together. The patient's history, without their name, we don't know identifications, their clinical history of treatment and how they progressed, and very, very importantly, what happened after the tumour sample was collected. Then the process starts of the sample collection or sample processing. So the tissues have now been collected, they've been sliced, they've been stained, and we've had a pathologist look at them. So a piece of those tissues, we call a section, goes into a lab for processing. And processing is basically digestion. So the principle of digestion is straightforward. It's the same enzymes in the human digestive system in our stomachs that breaks down protein for food. We harness those in very specific ways to break down the tumour sample into pieces, peptides, fragments of proteins. Now what we've got is a mess at that point, and what we have to do is clean it up. So we have to remove lipids, fats, connective tissue, DNA. We don't like DNA in these samples, just to get the peptides out. So we got that down to a fine art. It's very, very fast.
That's a new sound.
Yep.
Just cut for a second.
Then we're ready.
Okay, we have to do it again. That's it, 2 minutes.
I don't know how simple or depth you want. That's the point.
Oh, we were going great. The post, they're chatting in the background. They're having something.
Okay, time to stop making noise. Thank you. But keep doing it. Quietly.
Hardly. Like we crashed it. Process. Quick. You get the start with the
Samples coming in.
yeah.
Start again?
Yeah.
Okay. So
Let's do it.
why ProCan is so special is our collaborators, the teams of pathologists and researchers in 125 universities and hospitals around the world. They collect the samples of the tumour and the nearby adjacent tissue that's supposed to be healthy. They process it, and they have to collect the clinical records for those patients without the names. But we have to know the outcome of that patient, what they were treated with, and what the results were.
Well, how's this going to help?
Because what we're doing is comparing a group of patients, for example, they all say have colorectal cancer. Some of them survived treatment and some of them didn't. And we need to know what makes the ones who survived special. Those that didn't survive the treatment, whatever it was, we need to know what about their tumour can we predict to make use of for a drug treatment or an alternative drug treatment for that particular group of people. So we do that on a large scale first, like 200, 300, 500 cancer patients. And the goal is to get that down to, we know what we're looking for after the end of our discoveries. We know what proteins we're after. So can we apply that to the individual person? And often the answer is yes. And can we give that through the appropriate channels to a treating oncologist, so when the patient is in front of them, they can make a clinical decision on the spot with the ProCan data, along with all of the other clinical data they already have. Will the proteomic data change their decision-making process? And we believe the answer is a lot of the time, yes.
Because they've got something in common and you know its history. So you're helping the doctors and clinicians and oncologists out. Who's going to get the benefit of this? Obviously the patient. Who's going to use it the most?
Who's going to use the data the most? Data scientists will use the data the most, because it's just natural to find ways to use the data to discover new things and to come up with new tools. The data we generate will be made public slowly over time. As each group of data is published, we make it available to the world, and so other scientists will play with it, and play with it literally to see what they can do with it. Can they compare protein data collected in Australia from protein data collected in Europe, get meaningful information out of it. But the most important thing is it forms one giant knowledge base, a database we call the ProCan knowledge base. And that database currently is over 32,000 cancer samples in it. There's nothing like that globally on that scale, all collected the same way with the same method and the same quality all the way through. So from one set of tumours that may be colorectal to sets of tumours of all cancer kinds, all types that there are, that's going together. So the discoveries that will come from that beyond the treatment decisions that we're looking at now can be immense in the long run. So that's why I do believe data science is actually the most critical users of the data. But our goal, in ProCan's goal, is very specific. We want to use that data to design a test to be able to run it in a pathology or a registered laboratory where that data is clinically approved to be used and licensed to be used by oncologists and clinicians. So the end result will be oncologists anywhere in the world can send a sample to ProCan or a facility, run the tests that we're developing one by one, and get the results back within something like 24 hours to consider along with all the other clinical data they have anywhere in the world. So it doesn't have to be locked in a pathology lab or hidden behind doors. The information can be made available to clinicians directly. But there's a process to go through before we've got to that step, but that's our goal.
Ask yourself the question.
Yeah.
How did you do this before?
How did we do this before mass spectrometry went crazy? We didn't. So what historically has been done is by trial and error and by researching one protein at a time, people have discovered particular proteins that are increased in specific tumours. An example, the estrogen receptor and progesterone receptor in breast cancer, but not all breast cancer. And so over time, what people have done is develop antibodies for one protein at a time, so they can measure one protein at a time. And that's turned into current pathology, where a few hundred proteins over time have been accumulated in pathology labs, and they will stain the tumour from the patient with the antibodies to the proteins that they think are relevant to that tumour. So 1, 2, 3, sometimes 4 antibodies. The difference ProCan is making, we do 6, 8, 9,000 proteins at once in that tumour. So there's an enormous increase in the amount of information we have about that tumour over and above what's been done in the past, which is basically 1, 2 or 3 proteins at a time.
Is that how you started? One protein at a time?
One protein at a time is the history that we want to get past.
That was your history as well? I mean,
The way this is developed, I can just give the example of my own life story as a scientist. I spent 4 years trying to discover one protein in the brain. I spent 4 years in the US trying to isolate or purify that one protein so I could try and identify it. Then I spent 4 years postdoc in Melbourne trying to clone that one protein. And the day we cloned it was the day that we identified what it was, Dynamin. So that's 8 years, 12 years, if I do the maths right, 12 years of my life to characterize one protein. And that's how all science has been going in the 70s, 80s, 90s. One at a time, one gene at a time, one protein at a time. Mass spectrometry today measures around 8,000 proteins in every 20-minute cycle. So it builds on the knowledge created by the genome, the whole genome project, Human Genome Project, which we now have hundreds of human genomes sequenced. So we know what the protein should look like, but we want to know what the proteins are in the tumour. And being able to measure 8,000 at a time, every 20 minutes, it blows my mind. It's extraordinary. Having spent my career on one protein and trying to find the one gene that protein came from. And now I can identify my protein in all of these tumours that have nothing to do with the brain. It's everywhere. And we can figure out if it's got any role in any of the tumours.
what were you saying about Nobel Prizes in mass spectrometry?
Are we still filming?
No, he's probably just gone for lunch.
I don't know if it's still on, that's all.
No, it doesn't matter. Well, what we
So,
said about Nobel Prizes and
So the key,
you said somebody 7 or 8.
one of many keys that makes ProCam work is the technology behind mass spectrometry. And where it is today is remarkable. The mass spectrometers have had an evolution that's been incredible. So in around the year 2000, a particular kind of mass spec was invented called an Orbitrap. Today, that's been combined with another kind of mass spec called an astral analyzer. But what mass spectrometry in general goes back to World War II. And people were using it initially as part of the Manhattan Project to separate isotopes of uranium to get the enriched uranium away from the de-enriched uranium isotopes. And there were entire foundries of mass specs just to separate two atoms, two chemicals. So over the years, the technology to do what we do today has evolved incredibly. And there are 6, at least, Nobel Prizes awarded for various steps along the way of the science, physics, technology around mass spectrometry. And what we have today builds on 60, 75 years of physics, chemistry, and instrument design. And electronics. Electronics has gone from millisecond times for the equipment to send messages around to nanosecond response times. And that allows us to do more and more in shorter and shorter times. So the instrument that we have today is not the instrument ProCan started with. It's an instrument that does the work of 6 of the instruments from 10 years ago. It does the same work as 6 previous instruments in one package.
So you bought 6 machines. Is that 36 in the old money?
When we started ProCan, thanks to the ACRF very generous cash input, we were able to buy 6 of what was a cutting-edge technology at that time, so 8 years ago. And that technology was called DIA mass spec. But what it allowed us to do for the first time was to keep track of the amount of a protein from sample to sample to sample. Not detect, is a protein present or not present? That's important. But the DIA kind of mass spec allowed us to measure the amount in every sample, the relative amount. And that was one of the transformative steps in technology. The transformative step that happened a year or so ago was to be able to do that with 10 times less sample, so you don't need much, and up to 5 times faster, and twice the number of proteins identified.
Are these the same machines?
That's in 8 years.
These are the same machines?
The instrument in the background is one of the new machines. It's called the Astral, Orbitrap Astral mass spec. The older machines, which built ProCam, I have a boring name. They're called 6500 mass specs, 6600 mass specs from a company called SciEx. But we don't need really to go into that. But
No. Have they gotten cheaper? Who bought that one? I'm just interested, actually.
when we first bought them 8 years ago, they were around $1 million each.
Yeah.
And now those instruments, of course, are less, but the replacement instruments that everyone wants, including us, are closer to 1.5 to 2 million each.
Oh, they've gone up.
They've gone up.
Man.
The complexity's gone up, and the output is a quantum shift from what we had when ProCan started.
Yeah.
But this is important. All the data we've collected from the 32,000 samples, specimens that we've had all those time, is all still relevant. When we use old machines and new machines, when we compare different kinds of machines, we've got the same base data. We're just getting more or getting it faster. So everything we've ever done is incredibly valuable to us, and it's forming part of our knowledge base forever.
And accelerating at 1 million moles an hour.
It's accelerating. Our challenge is being able to build in the cost, or updating the equipment. When we were first interviewed by the ACRF for the original grant that got us started, one of the questions asked was the obvious one: won't this technology be out of date in 5 years? And the answer is yes. And now we can show that, that new technology has just blown previous technology away, but none of it is lost or wasted. It is extremely valuable. It has the same value it initially had. And we're making discoveries. And no one has started this initiative on this scale. There are many people with these mass specs, and they're using them for research on many, many things, including environmental toxins are measured in these kind of mass specs. But our singular difference is the focus we have to reach clinicians with the outcome, and our network of collaborators who are helping us find samples and get the right patient samples into our instruments over time. And that's gold. It's just magic. That's the secret sauce for ProCan. Not just the technology, which anyone can buy if you've got a spare $2 million, but it's the collaborative network of scientists and clinicians and pathologists who are supplying us with those samples and the knowledge to know what the questions are that we should be asking for each patient. So we need to know, is the tumour likely to come back, metastasis? Is the patient likely to decline quickly, recurrence? We need to know all those sorts of things, and it differs from cancer type to cancer type. So each collaborating group, hospital, university, they have specific questions that's front of mind for that group. What is the challenge with this particular cancer in front of us now? And that's what we address with them. But we're able to put it all together into a giant database as well.
Oh, we have got time. It's good.
So
Did you enjoy that so far?
I don't know if I'm preaching too much. What do you think?
I think everything you said has been great. Love the secret sauce.
Yeah, no, I've got you.
I think the longer you spoke, the more points you were hitting. It was a bit technical at the start, but the longer you went on, the more
Yeah.
clear it became.
So you were going to maybe later film at loading the mass spec and stuff? Or
Oh yeah, we're
let
going to
them-
load it. I mean, also there's graphs that pop up. You might want to explain what the bloody graphs do.
Do you want to go into the spaceship thing?
I love going to the spaceship thing. You know, sci-fi is my go-to area, and same as you. Foundation. Probably not, unless you want to explain the which is right.
Am I in the same spot now?
Perfect. There, that's right.
So you're going to do it tonight.
It's all right.
Okay. But not, yeah.
This feels my favourite.
Do you want to know about mass specs
I don't know
on
what
Voyager?
it is. Do you
And
want to tell me? Yeah, I do. Go on. It looks like
just describing how
turkey basin.
describing how a mass spectrometer works is incredibly challenging because there are so many types. But one of the earliest kinds is this. It's called a quadrupole. 1, 2, 3, 4. 4 metal rods. They don't have to be hollow. They can be solid rods. And what they do, there's a space in between, not the 4 hollows, but the dead center. What happens is when we have a sample, it can be a gas sample, it can be a chemical sample, or we use proteins. They're sent through there by charging them. We put a charge on the samples and it's sent through there, attracted to the opposite charge,
An electric charge.
electric charge, either positive or negative. We pick which one.
Just so we go, let's say electric charge.
So we put, the samples have to be ionized, they have to be charged, so they have an electric charge. Oi, oi.
Dylan?
We're recording again.
For,
Thanks, Andrew.
for a mass spec,
Just go to your left a little bit, please. Right, great, thank you. Just
For
off
a-
the top, I guess.
mass spectrometry goes back to the 1940s, as I said. But since the 1970s, over 100 mass spectrometers have been sent into space. 100 at least. So some of the most famous ones are on the spaceships, Voyager spaceships 1 and 2, that are now, the first one is outside the solar system. There are also mass specs on the rovers on Mars. There are mass specs on the moon. So what they're doing is sampling gases, and they're sampling nowadays, more sophisticated, they're sampling the soil to identify the chemistry or not proteins, the chemical composition of gas or of soil or of atmospheres. So those gases in space around on Mars are passed through the hole electrically. They're charged. And there are 4 rods in this case. There can be 8 rods. And there are 4 cylinders called quadrupoles or octopoles if there's 8. But the important thing is the space between them. Those ions, those electrically charged chemicals, are what we call flying through the centre of these 4 rods because they're attracted to an opposite charge at the other end. But the secret trick is that we're applying voltage variation to those 4 rods. So we apply charge to each rod differently through the computer, of course, in milliseconds. And that rod controls the field, the electrical field inside that space. And the consequence of that is the ions that are flowing through, the charged particles, we can control their flow. We can make them go this way, that way, or let them go straight. And if we don't want particular ions, we set them off course and they hit the sides of these rods and they're there until one day they may be cleaned in 100 years later. But the ions, or the item that we're trying to measure, flows through. Okay, then there's another mass spec.
What does that look like? Can we see that on... Can we see that in some way? Or is it
No.
just numbers?
No, so what comprises a modern day mass spectrometer that's not on a spaceship is a vacuum pump. So this only happens in a vacuum. That's easy in space. So all the noises mass specs make are mainly vacuum pumped to keep the atmospheric pressure out because they're open to the air at one end. And the samples go from one mass spec to another in a line. And if we want to make the path of these ions curve, we apply another charge and push the ions down by just like an ion mirror, force the ions down. So we can control where they go, all in a couple of nanoseconds, until they hit a plate. So the key principle is separation. Separate the ions one by their size. So two sizes of uranium, strontium versus whatever. In the case of peptides, one peptide mass versus another that is part of a protein. They have to be separated in here. So in biochemistry, we used to call it chromatography. It's a bit like that, for those that know biochemistry. So the goal is to make the ions have different paths and we make them slow down at different speeds and speed up at different speeds in the mass spec until they're separated and they hit a detector. The detector is just simply the opposite charge and detects current. So if one atom that's got one charge on it hits a detector, the detector literally goes ping, one record. If 100 hits, there's 100 pings. And with scale of nanoseconds and separating our flow of ions or charged particles by nanoseconds, we can detect the time at which they hit, and we can use mathematics to determine the mass of what hit that detector, whether it's an amino acid, a protein, whether it's a chemical.
Because everything's got a unique way.
Yes, and we can use the mass specs to control the mass flow. So we're selecting which charged particles are allowed to go through at any one time.
Yeah. Wow.
So you can't see it in action because there is a whole series of these in a row that do different jobs. The first one is mainly for cleanup, to get rid of the things we don't want in the mass spec. And it just gets more and more complex after that. I need a physics degree just to explain the newest mass spec at the end, called the Astral Analyzer. Yeah. No, I don't. I know what I want to do with these instruments. ProCan knows what we want to do with the instruments. We could spend our whole lives trying to improve them, but we wouldn't be using them.
Leave that to somebody else.
Leave that to the experts, the Nobel Prize winning experts.
Now, how many Nobel Prizes? 7?
At least 6.
6.
Yeah. I can look up the number for you exactly
No,
later. I didn't come
just
prepared.
Each breakthrough signalled another Nobel Prize, is it?
They're Nobel Prizes in physics and chemistry for the technique that was used, not just because it helps mass spectrometry, but it helps a lot of physics, a lot of mathematics. So they were discovered along the evolution of mass spectrometers to what they are today.
All from the atom bomb.
Yes.
Was physics
Well, for the
going
atom,
that way anyway?
for the atom bomb, it was part of the initial early days of uranium enrichment. The particular masses of uranium that they wanted in a reactor and particular masses they didn't want, because there's many isotopes of uranium. So those mass specs were the first mass specs in mass production, but they were largely purification system, because they isolated the atoms that they collected. We use such a tiny amount. We don't isolate anything. We just consume it on the electrical plate at the other end.
I'm seriously going off piece now.
Yeah.
The editor's going to get really annoyed with me. Just have to slug through all this and understand it. So, you put it in, it comes out, and you get mathematical numbers.
Yes.
It's not visual at all.
What comes out the other end of mass spec are graphs. So the samples are fed into a mass spec over time. If they're really simple, like gas, you just inject and read it. But we have incredibly complex samples, extremely complex, with millions of pieces of proteins, peptides going in at once. So what we get as a readout is over time, 20-minute run, we get to measure which masses came out of the mass spec at which time, and we get the dimension of how many hit the detector, intensity. So we've got these curves indicating one peptide came out here in 5 minutes and has a very high intensity. One came out at 6 minutes, has a very low intensity, and so on.
Yep.
And that gives us the total picture of how the whole mass spec run looks like. But the science is we zoom in on each of those curves, and there are many of them, down to each one signal, one at a time. What does that represent? That represents one peptide or one piece of a peptide. And we monitor how much that one peptide, throw away all the rest of the information and monitor one peptide at a time. And we can see how much there is by the area under that curve over time.
Peptides are the key.
Peptides are the key for us. So proteins are digested to small peptides. Peptides are the right size for these mass specs. They're much bigger than gases in outer space, but they're smaller than proteins. And mass specs don't work on a high throughput way with whole proteins. They're too big. So peptides are the right size. And we're trying to put Humpty Dumpty back together again. So we've taken a tumour, digested it with enzymes like trypsin to pieces. The pieces are peptides. We identify those peptides and we put those pieces of the peptides back together again and say what proteins were there.
Thank you.
That's how it works.
That worked for me.
I was worried about it still holding that
No, that's
for your
okay.
editing purposes.
We actually in 4K, we can go in close to the
Oh,
live.
cool.
What would be handy, I'm just going to zoom in now, what would be great is just to go through how you were saying, things go through that end there. And
It's hard to show with that. There's a second one I can use
well,
in the other
you've already
hand.
shot you with it.
Yeah.
It would be best to stay in continuity with
I
that.
might
Just gonna
have
make
a pen or a
positive
pointing
detail.
device would be easier.
Pointing
No,
thing?
I would really be best if you just did what you
Okay,
can.
fine.
We're going to have to cut it.
Yeah.
So, and I'm ready to do that when you are. So Stuart, I've just come in here. No.
Yeah, he doesn't want
Probably
it.
a very
It
bad
won't-
idea for continuity if you
No,
wanna
no.
go to-
But yeah, I would say it's a pen on the spot.
There we go. So I'm ready now to do that. Okay,
So the heart of some of the initial mass spectrometers, particularly the ones that are in spaceships, Voyager, rovers on Mars and lunar landers, are quadrupoles. So what we have are 4 rods. You can see the rods here. They don't have to be hollow, they can be solid rods, so long as they hold electric current. But what you can see down the center is a gap. There's a hole down the center. So inside a mass spectrometer, what's happening is the electrically charged particles are passed into the hole. Not the 4 hollow rods, but the hole in between the 4. Or there could be 8 for an octagon. And as it flows through, as the charged particles are flowing through the mass spectrometer, we are controlling the current on the tips of these metal rods. So we control it through the computer, and by applying different charges to each rod, we create a radio frequency field inside that gap, inside the hole. So as the ions flow through, they're being controlled by us. We are saying we don't want particular ions that are too small, so they're sent to the walls. They'll hit the sides of these rods and we'll clean them up in a year or so. The ones that we go through, a field that is designed to only allow particular masses through at a time, they go straight through to the next part of the mass spec and the next. So if we tune this, for example, to allow only an atom through that has a mass of 1, and only that gets through, the mass spec detects it. Then we tune, the sample is still coming in because we flow it in over half an hour. Then we tune this, the computer does, I don't. What's got a mass of 2, what's got a mass of 3?
I don't want to stop it. I think we're more than enough.
We're tuning it.
And that means I get Phil talking about something he doesn't know we're filming. And then we get the best result.
I don't know, I've got Humpty Dumpty and I don't know what else I put in there.
Yeah, Humpty Dumpty was good.
That was a good analogy. What else you got? You're just making doctors look good, we understand. What happens on the screens back there? What can we, is it something you should explain that comes up? What are they looking at?
Yeah, what they're looking at now is the tip. So the samples go into a machine before the mass spec, 100 at a time on a plate.
Yeah.
And then the robot picks one sample at a time through the tiniest tiny needles and injects it into the mass spec. And it does so over 20 minutes. But the needle at the end is where we put voltage and we make sure those peptides are charged. They're no use if they're not charged. So every now and then they get clogged, they get dirty, or the tip breaks. So you have to monitor whether there's smooth flow or there's a calcification on the edge of the tips. That's what they're looking at now. Yep, she's looking at a tip. You can actually see the needle.
Oh, I can see the needle. Moving as in real life.
So the metal circle on the left of that screen is the opening of the mass spec. It's equivalent to just before this hole here. And a mass spec is open to the atmosphere because we've got to spray things inside the samples. But the other end of the mass spec has to be super high vacuum. And so there's a series of vacuum pumps vacuuming down constantly.
Okay, you want to do off-the-cuff benefits again? Apart from, because you were 28,000 when we filmed you last time, you're up to 35,000.
It's 32 is the official number we use. We're actually about 35,000, yes. But I didn't look it up today.
It's okay, it's going to change.
It's growing.
Yeah. And that's from, how many different racial, across the full spectrum, I imagine?
How many?
Racial, does racial make any difference?
Oh.
It's around the world, is what I'm trying to get at, I suppose.
Australia's a good spot to do this study because of the racial diversity in Australia. We don't represent all racial regions of the world very well, but we're very, very broad. We hope that we'll be able to collaborate with groups in India and China to extend that. There's lots of ways that we can develop a racially diverse database. But right now, being in Australia, it is pretty good.
See, that was my assumption, but does race make a difference or not? Or we don't know?
Race makes a difference in some cancers.
This will probably never get in, but it's okay.
Race, I can give you an example of lung cancer for people who've never smoked, ever. So the rate of never smoking lung cancer in Taiwan is probably double most of the rest of the world. And it's not pollution, it's not smoking, of course, and it's not particular viruses that we know that can cause cancer. So that's coming down to racial genetics, and the basis behind that is not completely resolved yet. So you have pockets like that all around the world. That in India, where I was a few months ago, there are subpopulations, major racial populations there, several, where the clinicians have told me they do have particular cancer types that are much more prevalent in one population than another. So I don't have that data, but it's not universal.
Does anyone have that data?
No, we're all chasing it. It's not that prevalent. The vast majority of cancers are cancers, like children versus adults. Children get the same kinds of tumours as adults. They just get a smaller number of kinds of tumours, because they don't have prostate cancer, breast cancer, things like that, obviously. But the sarcomas and the leukemias they get are pretty much the same as an adult cancer. And race is similar to that. There'll be some differences due to race, but there are differences that are much less than what the commonalities are.
And kids have different reactions to cancer to adults, don't they? They fight it off in different ways, or?
Well, obviously children are still developing, so their bodies can respond and adapt much better. So I'll give you an example. There are certain leukemias in children that are essentially 90 to 95% cured by chemo and radiation therapy. And in adults, the same leukemias can be a 50% cure rate. So there are lots of things that differ between children and adults, but it's, in my scientific understanding, but without the exact knowledge, I think that's just the general flexibility and growth of a younger person versus an adult.
Is it the super hormones and everything else that's going through that possibly changes it? I don't know hormones.
No, this is quite correct, and I could go into that, but
Yeah.
what sustains leukemia so that it keeps growing when the chemotherapy is circulating and it kills the circulating chemotherapy, but certain leukemic cells survive because there are survival signals in different parts of the body, the spleen, the bone marrow, and those survival hormones keep that part of the tumour alive, so when the chemo stops, it just comes back. That's actually something my team is studying, how to kill that.
Well, no, that's what Belinda Parker was working on in Melbourne, Peter Mac. I think
Yeah.
she's got the bones,
Yeah,
and she basically said the bone marrow and the cancer cells. If the patient dies, the cancer cell dies.
yeah,
So in order to study them, she was trying to get the bone marrow to stay alive.
yes,
And she went to a butcher and got some beef bones, and then went to her brother-in-law, who was a professor in engineering, to come up with a pump that could actually keep the bone alive so the cancer survived.
yeah,
And
yeah.
they managed. Are you aware of that one? Or?
I'm not aware of the specifics, but it makes complete sense.
And successful. She said, well, that's been we can examine the cancer now.
So we've published a paper recently with a large collaborative team, including Canada, and we showed that the leukemia has stem cells in the bone marrow, and the stem cells can survive radiation and chemotherapy. And we have found a drug that you can apply to the patient mice while undergoing chemo that wipes out those cells too. It stops the protection that those cells in the bone marrow have. And I'm actually running a research lab, trying to turn that chemical compound into a drug for clinical use. It's a major part of my research project outside ProCam.
How's it going?
It's going very well.
Thank you very much.
Need funding.
And you're doing that here?
Yes.
Okay.
And in Newcastle, Australia, with the chemists, and in Canada with the oncologists.
We
So-
need you, go.
Yeah. What do you need now?
Well, explain what's happening with the machines, I suppose, before we lose you.
Jennifer, just let Jennifer stay, or is it how designed to stay?
Steve, 30 questions you want to ask us? Interesting. Tell.
There are different peptides coming out and they have different intensities. So how much they're detected by the mass spec is by the height. So, but if you go to the bottom curve, you can look at any 1 second of time, and when you do that, you see on the bottom trace the fragments or the actual peptides or pieces that are being detected at that 1 second in time. So the analogy I gave about Humpty Dumpty, these are the pieces, these are the shells of that egg, thousands and thousands and thousands of pieces that we have to put back together to say what is the identity of that peptide and what is the identity of the protein they came from. So on the screen at the bottom you're looking at, say, a main peak there at 4,000 Daltons or 4,000 atoms big. But there are many other peaks, and they all tell us the data that our computers deconvolve into identifying what is that peptide. And if you go back up one curve up in red, you can see there's thousands upon thousands of them in any one trace. So that's the data that we have to analyze to put together a proteome from one sample. And that's why we use machine learning, that's why we use computers. They do the calculation on that data for us. In the old days, we looked at those traces on the bottom one by one, and we tried to analyze their meaning one at a time. Nowadays, with 8,000 proteins per run, you can't do that anymore. There is just too much information there. So it's been automated.
So Jennifer, excuse me, I'm just going to get in there a little bit. That's all very good.
You like the cake, and here is the cake after it comes out of the oven. She's good at that. She's got much more value than that to Procan, of course, but
No, it's not the end of it.
I'm going to talk about needles and sprays and tips. What you're looking at is the sample actually being sprayed into the mass spectrometer. So on the right side you've got the needle, the syringe, and the sample is flowing out of there over a 20-minute period, and it's being sprayed, literally just sprayed at the mass spectrometer, which is on the left. The opening of the mass spec is on the left.
Okay, so we'll do that again. I'm just going to do a little thing here. Okay, and just talking about what happens.
So what you're looking at on the right, you're looking at the spray tip. So the output from the HPLC or the LC is spraying the sample through this spray tip over 20 minutes. So it's just continually spraying. At the very end of that tip is 2,000 volts or so to ionize or to electrically charge the molecules, which you can't see. And the spray is going into the mass spec. The grey round thing on the left is the opening to the mass spectrometer. It's open to the atmosphere, but inside the vacuum gets higher and higher as it goes inside the instrument.
Excellent. Now what I'll do is I'll get you to do that again, and I'll just get you to say it goes from the needle tip on the right hand side and is sprayed onto the mass spectrometer. Just that short statement. Okay, and action.
The ions go from the needle tip on the right hand side, sprayed into the opening of the mass spectrometer on the left.
Lots more of that. Okay, and ready.
The sample is sprayed through this spray tip over a period of 20 minutes directly into the opening of the mass spec on the left.
Excellent.
So the instrument we're just looking at is a discovery machine. It measures thousands and thousands of proteins, up to 8,000 in a 20-minute run. But if the same sample, exactly the same unchanged sample, is put into this instrument, this class of instrument measures targeted proteins. We program the instrument to only measure the amounts of the proteins that we're targeting. And that target list allows the mass spectrometer to ignore all the other peptides, ignore all the other data, but just focus on the data that we want. And the outcome is one sample, one patient, we can record the exact amount of that protein, many of them at once, in that one patient. And that's most important part of that is that's what will be in a clinical use in the future. That's our goal, to place instruments like this one into pathology labs where they already use similar instruments to have the actual test done on the sample for one patient at a time.
You said it really succinctly. First one, you said, that's for that, and this is clinical, and then you got excited.
This machine is for discovery and finding the biomarkers, and this machine we're looking at now is to turn those biomarkers into a clinical test in a pathology lab in the future.
This is the hero?
That's the hero. That's the outcome of all of the work and all the thousands of samples. We'll be measuring one patient and how much of particular proteins are present in that one patient. Because that's the data we hand to a clinician or an oncologist. We don't hand them the data on 8 or 9,000 proteins. It's just too much information for a clinician to make any use of. They want to know, where's my estrogen receptor, where's my progesterone, where's my markers, how much of those? And this tells them how much there is in one sample, one patient. It doesn't rely on 100 or 1,000 patients like the other instruments do.
Why does it look so dull?
Why does it look so dull? I don't know. It's a
It's not important.
SCIEX grey. The magic is inside. If we open the box and look under the hood, it's amazing in there. It's just a jungle of wires and tubes and various mass specs. But the two technologies, they're both mass spectrometers, but the principle here is a completely different principle. So they're different kinds of mass specs in a different order. So they're actually called triple quadrupoles because there's got 3 of those quadrupoles I showed you earlier in a row, and this doesn't.
Stand added by lights.
This is
You
our
can
path.
speak loud.
This is our
Come
path to
over
the
here,
clinic.
it's safe.
On the light, come over this side. That's the lighting this machine.
Just a sound recorder.
Okay.
Well, this is the big ones.